Okay, so today we're learning about the action potential. And so, based on the simulator, you should have discovered that neurons are like the dominoes or domino effect, in the sense that when you push the dominoes that are already set up, the energy that passes through them is like a wave. And so, once the first one falls, the rest will then fall in succession. So it's kind of like an all or nothing approach with dominoes. Just like dominoes, the action potential is like so. So neurons are like the domino effect. The protein channels are all set up, and once the first one opens, the rest open in succession. This is an all or nothing response. The wave travels through the dominoes like it does a neuron. The channels are then reset so that the neuron can react and fire again. And so there is a time that the neuron has to reset. It's called a refractory period. And in order for us to, kind of pause on that, that takes time for it to reset, and then we can fire again. Over here, this part of our notes is talking about the types of ions we have when we're talking about action potentials. So the first kind is the anions, and anions are going to be our negatively charged ions. So they are concentrated more within the cell. So think in. Examples of anions are chlorine ion and charged amino acids or proteins. Those garner a lot of negative charge, so they are going to be what's driving the whole force of the negative charge inside of a resting neuron. So that was one of the questions on the simulator. What is missing from our simulator that is a very large negative charge? And the answer is amino acids, or the negatively charged proteins. So in our picture, there are these large orange blocks here. On the flip side, our positively charged ions are called cations because they're positive, like cations. That's how you can remember it. I think that sometimes chemistry teachers teach it that way too. So cations are positively charged, so they are going to be more concentrated in the extracellular environment or fluid, which is outside the neuron. Examples are going to be sodium and potassium. So you can see this is outside the neuron, and this is in, and at rest, these two are the way they are. So you can see it says overall charge is positive at rest outside, and overall charge is negative inside. And then you can see the concentration gradients of each ion. So you're going to have more of the sodiums outside of the cell, more potassiums inside the cell, and chlorines are more prevalent outside the cell. So then you can imagine what direction they naturally diffuse in, because we already defined the word diffusion in our last lecture as the movement of particles from high to low. So at rest, potassium channels, which are more plentiful in quantity than sodium ions, at rest many of them are open, so we call them leaky channels, because then the ions just kind of leak through the membrane. They don't have to be going on each direction based on something. They're just leaking through. So based on the figure, which direction that potassium will diffuse. So it looks like to me we have a lot of potassiums on this right side and barely any on the left, right? So I think we have one over here. So naturally, according to diffusion, they're going to move out of the cell. And it says, what does diffusion even mean again? We already defined it earlier in our last lecture. Like I said, it's the movement of particles or ions from high concentration to low concentration. Okay, sodium channels at rest, most of them are closed. And based on the figure, which direction will the sodium diffuse? If it has the possibility to, it'll diffuse in. Okay. So we're basically setting up for the resting potential, which, if you found out in the simulator, is when the neuron is not being excited. It's not being stimulated. It's just at rest. So one thing about the membrane here is that it's polar. That means that there are opposite charges on opposite sides of the membrane. So the charges are dictated from this picture here, right? This idea that we have more positively charged items outside of the cell at rest, and we have more negatively charged things inside the cell. And by things, I mean the amino acids are really driving the negativity here. Okay, we have a lot of potassiums, but they're really small in comparison. So the idea that one side is positive and one side is negative creates this charge gradient, which it creates this thing called potential energy or stored up energy, like a battery. Okay, this electrical energy can be measured hypothetically with a voltmeter, because remember we're dealing with not chemical energy, but this electrical current, we'll say. So a neuron at rest, or it's unstimulated, has a resting potential of -70. We discovered where this number came from in our simulator. So you guys did the math. You used the Nernst equation to figure out this value. Okay, roughly. So this value, or this neuron resting potential of -70 millivolts, is maintained by the leaking of potassium as well as the sodium-potassium pump. This pump, if you recall, is an active transport pump. Okay, so sodiums and potassiums are pumped against the grain, or against, we'll say, their charge gradient, to reset the neuron once it's been stimulated. Okay, so, and that resetting process gets us back at a unstimulated or resting potential of -70. So right now is a good time to pause this recording. Okay, take a pause and click on this video here to have a nice little review of what I just spoke on. Okay, now we're going to talk about the nerve impulse, so the action potential and how that starts. So what happens is you get a stimulus from the external environment, or maybe even the internal environment. So a stimulus has to happen, and the receptor neuron has to detect that stimulus. Okay, so the nerve is stimulated. So by light, heat, or even a signal from somewhere. So if the stimulation is large enough, and what I mean by that is if it is above a certain threshold, the action potential will trigger from a special place on the neuron right here. It's like right at this spot here called the axon hillock, okay? And what will happen is the sodium channels will open in the cell membrane of the stimulated neuron. The sodium ions diffuse into the cell, causing the inside to depolarize, which means it's going to become more positive inside, right? And that's above threshold, and then that makes it go. Okay? The charges will flip or reverse at this point on the neuron when the sodium channels are letting sodiums in, because sodium is a positive charge. And we know at resting, the membrane is a minus or negative charge, minus 70. So all this flood of positivity of these ions are coming into the neuron, and that's gonna, we call it depolarize the neuron, okay? So the cell becomes depolarized. So you can see here in this image what I'm referring to. So this part in pink is when the neuron is at rest. It's not being stimulated. But once it's stimulated and it meets threshold at this axon hillock, then the sodium ion channels are going to open, and those are going to flood in with those sodiums, and it's going to depolarize the neuron at that point. So if this was the action potential coming down the axon, because it is one direction only, you could see that the wave of the depolarization, right? This idea that the sodiums are flying in, and it's moving this way, just like your simulator was moving towards you, it's the same concept. And then after, we got to kind of reset, what we call repolarization, to reset the neuron to be ready to fire again, okay? So this picture up here is just showing, okay, we have a stimulus, the receiving neuron is going to take that information, it's going to propagate, okay, that sensory neuron. We usually have a unipolar neuron there, except, a couple exceptions. Then that information is going to, turn into a chemical signal and relay to the relay neuron or interneuron. It's going to change into a chemical signal again, relay to this motor neuron, and then the motor neuron is going to take that electrical signal and then do something about it and trigger an effector like a muscle or a gland to move your hand or do the things that you need to do to get out of harm's way. Okay, so like I said earlier, the signal is one directional. The flow of potassium out of the cell stops the activation of sodium channels in the wrong direction. So what we mean by that is, because it's starting to repolarize here, there's no way of going like backwards. You can't, the sodium channels will not open in the wrong direction. So it's only a one-directional mechanism. Okay, so I mentioned that there were leaky channels earlier up here. So I said, hey, we have these two channels, sodium and potassium channels, and they are leaking or closed during rest. And those are just normal transport proteins that allow those ions to move and diffuse freely. Well, we have these other types of channels called voltage-gated channels. So they are dictated by millivolts or charge differences. So the differences in the charge is going to open them. So if you take a look here, the channel is closed when we have one positive and one negative end. And then when the flow of the sodiums is happening and the charge, the wave is coming to that part of the neuron, the channels will open because their charge, one side of the charge changes, and like charges repel, and that forces open the gate. So the gates are only dictated in opening and closing based on an action potential arriving there. Okay, so we'll write that down. The ion channels open and close in response to changes in charge, as we know as voltage across a membrane. So we saw this in our simulator. Once again, I asked you, what's happening to these sodium and potassium channels, these voltage gated ones, the ones that look like they have like a little sphere stuck in them. And then when they close, the sphere comes out. So let me go ahead and show you what I'm talking about. So I have this here. I'm going to go ahead and zoom in. I'll stimulate neuron, and let's watch the red and the green. So the red and the green open and close based on the voltage arriving. Okay, so just a little reminder that the sodium channels open quickly in response to depolarization or that charge flip, and close slowly. The potassium channels open slowly in response to depolarization and close slowly. And so that the reason for that is that the potassium is kind of involved with the resetting of things. So the fact that they are slower than sodium there's a reason for that, so they don't depolarize again too fast, and that's a one-directional movement. So once again, I would pause and I would go ahead and play this video here to give us more information on voltage gated channels. Okay, so these pictures once again are reviewing what's happening. So when the action potential arrives at the spot, the sodium is rushing in, causing this depolarization where the charge inside a neuron becomes positive, and then it kind of spreads down the axon. And so once it's already been there, we'll say, so this part here has already kind of been through the action potential. That's called repolarization. That is, the sodium channels are closed and additional potassium channels are open, so that the membrane cannot depolarize again. And then we're starting to become negative again because the sodiums are now leaving. Okay. All right. So here is a graph of what this looks like. The action potential, starting with resting potential. So resting potential, if you look at the graph, is at -70. So a stimulus from a neuron or 2 or 5 has to happen, and if it exceeds threshold potential of -55, so it has to reach or exceed. So it can be -55 millivolts, -54 millivolts, but if it's -56 millivolts, it will not fire. So it has to be -55 or higher, or more positive, we'll say. Okay, so once that happens, where we get above where we should, it's gonna go ahead and do the all or nothing thing. If it does not, if it's -56 or more negative, it'll just go like this. It'll just fail and just, not happen. So what happens is when the wave gets to the part of the neuron, it's called depolarization. So this part here. And so that means that the sodium channels are opening, okay, and the potassium channels close during this time. Okay, so at that point, our membrane is our membrane is positive inside, depolarization, and that is the action potential arriving at our spot. Okay, so then so I'm going through the steps here. So step 4, oh gosh, I don't know how to get rid of this. Okay, at step 4, the sodium channels are starting to close and the potassium channels are opening. And so that is going to allow our membrane to become more negative again. And that's called repolarization, because if the potassium channels are opening, the potassiums are going to flow out of the neuron, causing it to become more negative again. So that's called repolarization. at step 5, which is over here. That will help reset the charge gradient because you want it to be different and you want it to be negative inside. 6 is saying, oh, oops, the potassium channels are closing slowly, so we actually became too negative inside. So this is called hyperpolarization, when inside the neuron becomes too negative because the potassium channels are closing really slow, so too much flew out. Okay, so this picture right here is just rehashing what we just talked about. The change in charge opens, next sodium gate down the line, those voltage gated channels. The sodium ions continue to diffuse into the cell, causing depolarization, and the wave moves down the neuron. That's called an action potential. Since the potassium channels are opening more slowly than sodium channels, the potassium ions, my words. Potassium channels open more slowly than sodium channels. Potassium ions are diffusing out, and the charges reverse back at this point, and that is called repolarization, to reset. So on the sim, there was a part where you clicked stimulate neuron, and then you let it happen. Let's go ahead and just show it. So we're stimulating the neuron, we did, and then I'm gonna go ahead and stimulate it, fast forward, go, stimulate. And you see how I am trying to click this button, this stimulate neuron button, and I cannot do that because there is a time where it has to reset. Okay, so that is called the absolute refractory period. It is the time period where a second stimulus will not initiate an action potential, and it lasts about 1 millisecond. Okay, it cannot initiate another action potential, okay, because everything is not reset yet. I don't have the ability to do that because my neuron is not negative yet inside. So at any time after that, before we can fire again, is called the relative refractory period. So that is the time period after the initiation of the action potential that requires a stronger than normal stimulus to initiate another action potential. So 3 to 4 milliseconds. And this is because the potassium channels are still being open because they close so slowly. Okay? And in your textbook, in the chapter 9 book pages, there's a spot in your book pages. Let me go find it real fast. Where it talks about neurotransmitters. And there are a couple neurotransmitters that will lower the threshold. I don't know where I went. Darn it. I remember where it went. Here's the neurotransmitters. But there is a part where it tells us, oh gosh, I've lost my marbles. Here it is. Caffeine in coffee, tea, and cola drinks stimulates your nervous system activity by lowering the thresholds of synapses. As a result, postsynaptic neurons are more easily excited. Antidepressants will keep the neurotransmitter serotonin in synapses longer, compensating for the little understood decreased serotonin release. So there are factors here that will affect these things, okay, these refractory periods and the threshold of minus 55. So just wanted to keep your minds open to that information. Okay. So how do we reset? Well, we basically have to get the sodiums back out and potassiums back in, and that's the problem we have, right? So both of those two ions are moving against their concentration gradients, so we need a pump to pump them back to where they should go, and that's where our good friend sodium-potassium pump comes in. So this is an active transport protein that requires ATP in order to pump 3 sodiums back out and 2 potassiums back in. And that gets us to reset the membrane, ready to fire again. So I would go ahead and once again pause, watch the clip on sodium potassium pump. I believe it's the Amoeba Sisters, which I love them. If you'd like to watch them now, please feel free to do so. Here is the picture of the sodium potassium pump. Again, I also have this in our cell transport lecture. The myelin sheath. The myelin sheath play a role in how quickly a signal travels. So a myelin sheath, if you recall, is when an axon is coated with oligodendrocytes or Schwann cells, depending on which part of the nervous system you are in, right? And so the myelin sheath is going to insulate the axon, the neuron. It's going to also speed up the signal, because what happens is when you have a myelinated axon, the signal will not travel the length of the axon, but rather hop to each node between the cells. So take a look at a myelinated axon. You'll see that the signal is not going all the way through like this, but rather it is hopping the nodes. And so actually the speed of the hopping is much faster. So I have the statistic there that it is 330 miles per hour versus 11 miles per hour if you were going to travel the whole length of an axon. So the hopping part is called saltatory conduction. Saltatory conduction comes from the Latin word saltere, which means to leap, dance, or jump. So that increases the conduction velocity of your action potentials. So if you are suffering from a disease like multiple sclerosis, the T cells of the immune system attacks your myelin sheath, resulting in a loss of signal because it takes so much longer, so much faster, and it is just such a trip to get those signals where they should go, and people with multiple sclerosis have a lot of troubles with that. So once again, pause and take a look at my quick clip on saltatory conduction so you can see how much faster it is compared to an unmyelinated axon. All right, friends, we are almost done here. We're going to finally get to the end here, the end of the axon we call the synaptic cleft, where the signal converts from an electrical signal to the chemical signal to move on to the next axon. So at the end, that is where the impulse must jump. We call that the synaptic knobs of the axon terminal. So what happens here, let's go ahead and look at the picture, is that the signal is arriving here at the end, at the synaptic terminal or the axon terminal, at the synaptic knobs, okay? So what happens with that is the action potential is depolarizing the membrane at the very end, which causes the calcium channels to uptake calcium. So this comes back to our original lecture on cell transport, right? So calcium uptake forces the neurotransmitter vesicles, which are these things here, to release neurotransmitters via exocytosis across the cleft to our postsynaptic neuron. So this, my friends, is the presynaptic terminal, the presynaptic terminal or presynaptic neuron dumping its neurotransmitters to the next postsynaptic neuron, okay? So in this case, this postsynaptic area is actually an effector, like a muscle fiber. So we're going to be stimulating a muscle tissue, and that's why we have acetylcholine. Acetylcholine is going to be a neurotransmitter that will tell muscles to move, okay? So that's an example there, one example. So the neurotransmitter is going to diffuse from high to low across the synaptic cleft. They're going to bind to protein receptors of the receiving neuron. So here's the protein receptors. And those ion-gated channels are going to open, and the neurotransmitter is going to be either reabsorbed or degraded. So some examples of a neurotransmitter that we might see are excitatory, which means that they are going to be more likely to cause the action potential to fire. So that is called EPSP, excitatory neurotransmitters, we're going to say. So an example of this is glutamate. This glutamate causes sodium channels to open and sodium to rush in. On the flip side, we have inhibitory types of neurotransmitters, IPSPs, those are going to be ones that will decrease the probability of a action potential to fire. So like GABA causes chlorine channels to open, chlorine to rush in, and that causes more negativity because of this minus charge, right? So more negativity means, hey, let's keep that negative 70, or even more negative, versus a positive charge being like, hey, let's be more positive and take that threshold higher than minus 55. Okay. So add them all up and you get a graded potential, and you'll see if the action potential might be triggered at the axon hillock or not. So that means, hey, will the action potential trigger if the summation of the EPSPs, or the excitatory neurotransmitters, is high enough to overcome the summation or the addition of inhibitory types at the axon hillock. Acetylcholine is a type of neurotransmitter. I want you to know that one's the one that stimulates skeletal muscles. So you have an enzyme called acetylcholinesterase. It ends in ACE, so it's an enzyme that breaks down acetylcholine, which is a good thing, because then it is used, and so on and so forth. So there are such things in life called acetylcholinesterase inhibitors. Those things are bad. Okay, so those are called neurotoxins. They block the active site, so acetylcholinesterase, the enzyme, cannot bind to acetylcholine and help that neurotransmitter be broken down and used in the muscle tissue, so that this causes paralysis in muscle tissue. Examples: this acetylcholinesterase inhibitor is found in snake venom. So when a snake bites something, it paralyzes its victim so that it can eat it while it's still living. So that is the venom has acetylcholinesterase inhibitors. Sarin gas is a biochemical warfare type of solution that has been used on people to kill people. Sarin gas has acetylcholinesterase inhibitors, as well as insecticides or other type of paralyzing agents. Okay, so the not so good part of anatomy is some of this stuff. Okay, so epinephrine and norepinephrine are also types of neurotransmitters that have to do with fight or flight. Dopamine affects your sleep, mood, and attention type of neurotransmitter. A lack of dopamine in the brain has led to diseases such as Parkinson's. Excessive dopamine might lead to a schizophrenic patient. And serotonin is also widespread in the brain, just like dopamine, and it affects your sleep, mood, attention, and learning. Last but not least, here I have come to the end where you might have this thing called a graded potential. So what this is here is your neuron that is going to be passing on a action potential if possible. You have 1, 2, 3, 4 different types of neurons synapsing with this one neuron in the middle. Okay, so you're trying to figure out, will the action potential fire? Will it go and pass on its signal to the next neuron over here in gray? Okay, so you're going to see 1, 2, 3, and 4. They're going to synapse with the dendrites of this axon, and we are going to see, are we going to meet threshold at this axon hillock part to then fire and move on. So once again, if it is not above minus 55 or more positive, it will be considered a failed initiation, and it's just not going to work. So we have 4 different neurons here, 1, 2, 3, 4. And so signal 1 is gonna have plus 25 millivolts. So signal 21 here, signal 1, oh my gosh, I'm tired. Signal 1 is plus 25 millivolts. Second is minus 10 millivolts. Third is plus 40 millivolts. And 4 is minus 5 millivolts. So first, my question is signal 1 going to be an IPSP or EPSP based on our previous definitions here? So plus 25 is going to increase the probability of the neuron generating an action potential. So it's going to be an EPSP, an excitatory neurotransmitter. Minus 10 lowers our probability of a action potential to fire, so that's an IPSP. Plus 40, EPSP. And minus 5 is an IPSP. So I have a collection here of EPSPs and IPSPs. So I got to do some math to figure out, will the action potential fire? And the rule of thumb is you have to always add them up. So we're going to add these all up. So give me a total in millivolts. And you will always compare or add to minus 70, because that's where your resting potential is. That's where your baseline is. That's what you start at. So minus 70 plus this number that you've calculated here. And that is going to be your answer for will the action potential fire for this graded potential. So minus 70 is resting plus the addition of 25 minus 10 plus 40 minus 5, which you should get plus 50. So minus 70 plus 50 gets you minus 20 millivolts. Minus 20 is right here. Let's see. Minus 20 is right here. Let's see here. I'm just pretending. So minus 20 is more positive than minus 55. So yes, my friends, it will fire. Yes, we will have an action potential. Okay, so the action potential will originate here and will propagate that way. Thanks for watching. See ya.